Given, and I'm a honeybee researcher and breeder at Purdue University. So I've been engaged in honeybee breeding now for about 27 years. And my passion is honeybee behavior and trying to find solutions to improve honeybee health. There's really no silver bullet now. So there's a few traits in the box that are out there, and I'll be talking about those. And then, of course, I'll talk about our breeding program. So for me, honeybees are, there's a lot of challenges. Like now we're having a lot of mortality issues in certain parts of the country. So for me, it's kind of like a death by 1,000 cuts for honeybees. So there's really no exact one cause, but it's a safe bet that if you take varroa destructor out of the equation, maybe the propensity for illness in our bees or mortality would be less. So I'll get started with this talk. This is one of my favorite talks to give. This is breeding the bee that bites the mite. So we've been breeding bees for years here at Purdue University. And in the beginning, we just basically selected on colonies that grew the fewest mites or low varroa growth. And then we kind of morphed into where we're at today. So this is like, any other behavioral trait. There's not a lot, as I mentioned, like VSH, for example. This is also an important trait. And we'll see how this progressed through our breeding program. And hopefully, this talk might inspire some other people to start selecting on this trait also, because like VSH also, it takes a lot of work, a lot of time, a lot of resources. So for any breeding program to be successful, you have to constantly select. That's kind of the take-home message with this. So I'll get started. See if I can get this to advance. Here we go. So first of all, you got to ask yourself, why breed bees? So it could be for overwintering ability, spring development. We want bees that build up quickly in the spring, so we can have large populous colonies that produce a lot of honey, or can produce many individuals to pollinate. Temperament, we want gentle bees, reducing of swarming, reduce or increase of the use of propolis. In the recent years, we're finding that propolis is very important also to have in our colonies. So having sticky bees is a good thing. Even a simple character as color. So color is important for some people, or for the purest type. Some people want the light bees, blonde bees, dark bees, whatever, or decrease the incidence of diseases. And of course, resistance to varroa destructor. And then there's other personal reasons. So breeding versus queen rearing, there's a difference. So most people here in the US, for example, or in the world, are queen rearers. So they produce many individual queens. So the idea behind this is to produce superior queens. So we got to have the absolute best queens to execute a honeybee breeding program properly. So most people are queen rearers. So there's a lot of people that are rearing queens that will say they're queen breeding, when in fact they're queen rearing. So there's a difference. So on the left here is an example of like a pedigree showing a different lines, different Italian, Caucasian, Africanized, whatever. So with bee breeding, you need to have a goal as far as a trait that you're selected for. So something as simple as color. So you got to have a different characters that you're selecting for. So with breeding, you're selecting on certain traits that are desirable for you, versus queen rearing, you're just producing many superior queens ideally. So it's essential to have the best queen for a breeding program. So this will make everything much easier. I mean, if you have the queens that you can produce, say from larvae that are like under 36 hours or less, because they'll have more the maximum number of ovarials. So the number of ovarials correlates to the number of eggs that she can lay. So it's important having, the best possible queens for a breeding program. So here is an instrumentally inseminated queen. This queen here was about 4 years old before it died, that we had years ago. This is one that my great friend and mentor, Greg Hunt, the honeybee geneticist, here he inseminated this queen. So a very beautiful queen, and you can see this retinue around her. So that when you see a retinue, some retinues are farther, some are closer to the queen. So this one's, she's displaying a nice invisible chemical uniforms for the queen, for the workers to respond to. So that's a good thing. So the fundamental principle of honeybee breeding or any breeding program, plants, animals, bees, is the offspring need to resemble the parents. So you need to see similarities. So these phenotypes, as scientists call them, are something simple as temperament and the color. So you look at all these bees here in this picture, they all look similar. So ideally, you want to see similarities in these characters that you're selecting for. So some steps in breeding. So you need to decide on the trait or the phenotype. And then from that, you need to develop an assay, then measure the foundation population, and then select the parents. And then you control the matings using a design. You want to evaluate, document, and verify, and select parents. So the key is you have to be vigilant, extremely observant, and it requires being in the colony, going through all the combs, and looking at all these different parameters, because there's a lot of mechanisms involved in breeding that are important. So I'm not going to talk much about varroa destructor. We all know that varroa destructor is basically, it's the #1 factor associated with colony mortality globally. And recently, as many of you know, this parasite is now in Australia. So hopefully we can help them out by importing stock from the US to kind of speed up the response to this parasite. So it's still causing a lot of problems, and so it's the #1 problem, really. Of course, American foul brood, we don't want that in our colonies, so that's pretty bad. So this I won't talk much about. So varroa destructor, where did they come from? They came from a cousin of our honeybees, Apis mellifera. So they came from the Apis ceranae. So the Apis ceranae has lived with varroa for thousands of years. So they've learned to live together in a way that it doesn't cause mortality of the colony. So, for example, the varroa only reproduce in the drone cells, and Apis ceranae bees are also excellent groomers. So through time, they develop really good grooming behaviors. And I might add that Africanized bees are also excellent groomers. So that's one reason they don't have a lot of problems with varroa. They also have a high propensity to swarm. So they came from these Asian honeybees. So surveys from the last decade of colony losses, basically, as I mentioned, most of it is associated with varroa destructor. Yes, there's other factors. We have loss of habitat, modernity, pesticides. Like in recent years, for example, all of our fence rows are gone. I mean, when I grew up as a kid, before varroa even came, we had all of our corn and soybeans had fence rows. And that was a really great habitat for not only honeybees, but our native pollinators. So all that is gone. And then we have these monoculture environments that also isn't good. And then, for example, taking many, 1.5 million colonies, for example, to the Central Valley every year. So this is a monoculture. So, and you have all these individual bees from different parts of the country. So it's basically a cocktail party for disease and parasites and viruses. I mean, the viruses are mutating, the mites are changing their behavior as I speak. Everything's changing, but this creates it, it speeds it up and to a negative force also. So it's very difficult when you have all these problems and then you have all these colonies in one area and then they come back throughout the United States. So you disseminate these more virulent mites, so to speak, and virus in some cases. So many behaviors are influenced by genes. So this is something I'm interested in. A few of them is stinging, pollen foraging, undertaking, hygienic behavior, etc. Even learning. So there's a lot of behavior. So some behavioral traits, there's not a lot because they haven't been as honeybees, our honeybees, Apis mellifera, haven't been associated with the varroa mite that long. So we have some traits like general hygiene, and we have varroa sensitive hygiene, VSH, and then we have grooming, we have mite biting behavior. These are traits that have been selected on. So there's a freeze kill brood assay. So there's two assays. One is if you don't have liquid nitrogen yourself, you can cut a portion of your comb out and put it in your freezer overnight and then reattach it. And then the bees will improvise the comb back and they will pull the dead pupae out in a certain amount of time. Or the classic liquid nitrogen method. So that's where you use an improvised soup can or something, or a piece of piping. You put about 250 mils of liquid nitrogen in there, but not everybody has access to liquid nitrogen. So just cutting a piece of comb out sufficiently. So the idea is if you use liquid nitrogen, you take these cylinders. So this is a comb of brood from an instrumentally inseminated queen. So we would test periodically just to see how they respond under general hygiene. So ideally in 24 hours, we like to see 98% or more removed. So this was how much they removed from that. So that was one thing that we also look at on our breeder queens. So if we test, say, 10 queens and one expresses this more, maybe that'll be the one we also use. So hygienic behavioral is a heritable trait. Walter Rothenbuehler found this years ago in Ohio. And so that was one of the first hygienic tests and selection that was made. And then we have VSH behavior, or the SMR as they call it, the suppression of mite reproduction. So Jeff Harbaugh and John Harbaugh, and Jeff Harris are the pioneers behind this trait. And later they changed it because they discovered a mechanism was different. So they called them VSH behavior. So it's basically an indirect attack on varroa destructor, if you think of it. And this would be like what we call this an additive trait. So there's several genes that are associated with this trait. And so, for example, you have one bee that'll come by, she'll scan the top of the cell with her antennae and she'll detect some anomaly. And then another bee, then she'll initiate uncapping. And another bee will come by and uncap. But then sometimes they make a mistake. Another bee will come by and scan the pupa and decide, oh, it's fine. There's no mite family or there's no anomaly. And then another bee will come by and recap it. So there's a lot of characters that are working in concert with each other. So we call this additive. Another example, I thought through this in here to show Rob Page for like 32 years. He selected solely on pollen hoarding. So he had what we call bidirectional mating. So you have these high lines and low lines. Totally out of scientific curiosity, but through this, many things emerged that were exciting and things that we learned about honeybee behavior and genetics. So here is an example of a frame that's in a high pollen hoarding colony. So can you have too much of one trait? Yes, you could have too much of a trait. So this is a colony that's in a single Langstroth hive, and you can see a little patch of brood there. So these colonies are not productive. This is totally scientific curiosity here. So now I'm going to dive into the genesis of what Greg and I started at Purdue with our breeding program. So this is Ernesto Guzman and Miguel Archuleta. So Miguel was Greg's first PhD student, and Ernesto's working at the University of Guelph now, and he's selecting on low varroa growth. So Miguel and Ernesto did a study in Mexico where they found that grooming behavior was important. And they found that those with the lowest infestation overall had low adult infestation. And they also had the most chewed mites on the sticky board. But they never selected on these traits. So here's the mites in a brood, so you can see between the low and the high group, they're very similar. But if you look at the mites on the adults on the low and high group, it's a big difference. So the ones that had fewer mites versus the high group, there's a big difference. So at Purdue, we started our breeding program in 1997, and we simply just selected for low varroa growth. The colonies that grew the fewest mites, that was the colonies that we bred from. And I might add, that is the simplest way to breed for varroa resistance. You can simply just choose your breeders from colonies that grow the fewest mites. So in the beginning, we started with a diverse group of survivors. We got the old SMRs, when we call them SMRs, and we got the VSH, we got the Russians. So we got VSH from Tom Glynn, John Harbaugh. We got 3 queens. And then we got Russians, because we know Russian bees, they're also good groomers. And then we looked at measured the mite population and strength multiple times. And then we also looked at, in the beginning, we looked at even the honey yield. And then we tested the best sources for VSH or hygienic behavior. And we also looked at the temperament. And then we also selected against general disease resistance. So colonies that, for example, we had chalk brood when I started early, and then we were able to breed that out within about 4 or 5 years. So when I made the nucleus colonies, my first nukes, for example, in the spring, several of them would develop chalk brood. Now, for when we make early nukes, we don't have a chalk brood problem. So it's easy to breed that out. And then in 2007, we started looking at grooming behavior. And so we started selecting on that. And then we started, after investigating grooming behavior, we decided to take it even farther, and we started selecting on mite biting or chewed and damaged mites. And then for a brief period, we looked at tolerance to viruses like deformed wing virus. There seems to be something there where they seem to tolerate the virus better, but we never were able to investigate that long enough to determine what the mechanisms were or what exactly was going on. And then strong overwintering ability and general disease resistance and calm temperament. Nobody wants mean bees. So the greatest diversity in a breeding program will happen at the beginning of it. So when you start your breeding program, you want to think about what traits you want and what traits you want for your area. So every area is different parts of the country. I mean, if you're in a dry climate or if you're in a really wet climate, so you gotta figure out what exactly is best for your area, because certain traits may not do as well in other areas. So again, we got Russians, we got VSAs, and we got some Ohio survivors. So Greg's brother Vince had a colony that he didn't treat or anything. He was just simply a hobby beekeeper, and the colony had like holes in it from woodpeckers, and so Greg got that queen. That was one of the early queens also in our breeding program. And they look like Caucasian bees, kind of these big gray bands on them. They were really beautiful bees. And then we have many diverse sources. So you should start to see more uniformity in your breeding program within, 3 to 5 years. So the hard work will pay off pretty quickly, depending on the traits. Certain traits are easier to select for than other traits. So the process for looking at grooming and mite biting is we insert these sticky boards and we spray them with vegetable oil. So we apply as much oil as you would a iron skillet to fry an egg. So you don't want too much, you don't want too little on there. We leave those in for 2 days, and then we transport them to the lab for inspection. And we try to do this 2 to 3 times a year, but most often we do this in the springtime and in the fall, so we can measure the mite population that way. And very seldom we'll have ants on there that are attracted to the vegetable oil and they'll eat it, like the paver ants, the little tetramorams, or the thief ants. I've had it where within 2 days they eat like 75% of the oil. It's incredible, but not too often. So that's the only, and very rarely that happens. Maybe out of 100 hives, you might get 3 or 4 that are like that. So the process, so I'm fortunate to have another grab that we hire every few years. We get a new lab technician, so they assist with this. So the process here is to remove the mites from the sticky board. We use a #1 artist paintbrush. So the oil on there, the vegetable oil, helps remove the mites, dislodge the mites from the board, and then we put them on these glass slides, and then we can evaluate them. So this is a typical sticky board. So we have these IPM boards that we use that are called Country Rubes bottom boards, if anybody's interested. They're located in California. It's a wonderful product, and they last about 12 years in our area where we're at. We got lots of rain, snow, cold, hot. So it's a really great product. And this is the mites here. So these little discs. So this is what we're seeing when we bring them in the lab. And then we place them on these slides. So each slide represents one unit, one colony. So as you can see, it's very time consuming and tedious. So it takes a lot of work to do this. So here's a nice view showing the mite being picked up with the artist paintbrush. So this works really well. What's nice about it is you can remove them without damaging them. So you got to make sure that the mites aren't damaged during transport from the apiary to the lab, and also when you're removing them, because that could give you a false positive. So looking at individual bees for grooming behavior, this is a picture showing an arena where we do these, what we call surrogate cage assays. So you got to bring bees into the lab, you apply mites to them, you see how long it takes them to respond to grooming. They need to do this with many individuals, and then hopefully it correlates with the colony or the breeder colony that you're wanting to produce more progeny from. So in some cases, this may take hundreds of individuals to really verify where you're at. So I got a video here. This is 2 individual mites from 2 separate instrumentally inseminated queens, and a varroa mite has been placed on her thorax. And you would think, a varroa mite is a really large parasite. It's like having something the size of your fist. That's about the size. on your body. If you take your fist and put it, that's how it's a large parasite, and you think they would respond to it quickly. And another thing to think about, all this that I'm talking about is happening in the darkness of the hive. So they're responding by the movement. There's a lot of unknown, there's a lot of things we don't know about this trait still that we need to learn. So you have these two individuals. So you have this classic windshield wiper motions. See how she's trying to dislodge that mite, where the other one, there's absolutely no response at all. So you need to test many individuals. So the one that scores the highest as far as the grooming, that would correlate to the field, then that would be a breeder queen, for example. But this takes a lot of work and time to start, but it's worth it once you do it. Let's see if I can go next. There we go. So these are normal looking varroa mites. So you can think of varroa destructor as a dirty hypodermic needle. It's a vector virus. It's vectors. It's not the mites. It's not what you see that's causing a lot of problems. It's what you don't see. It's the viruses that it's vectoring, like DWD, for example. So the varroa mite is literally chewing into the honeybee, the workers between the segment plates, tissue, soft tissue that she's feeding on. So she's feeding on the fat bodies and the hemolymph. As she's doing that, she's introducing virus to the bees. So it compromises the individual bee's immune system, but as a complete colony, you can think of a honeybee colony as a circuit, or we can call it a organism. So each bee is connected so that they're talking to each other, they're feeding to each other by trophallaxis. So it's easily disseminated among the whole colony. So that the whole colony immune system is restricted or depressed. So it suppresses all of this through time, not just the individual, but the whole colony. So, and the more individuals you have in the colony, the more mites present, the more problems you're going to have. They're going to be amplified. Because like DWV, for example, it was around. We had a lot of these viruses. They were around even before varroa came here. It's just the bees never had any problems with it because they didn't have this vectoring parasite. And the viruses are mutating also. And something that's not talked about a lot is the mites. The behavior of the mites is also shifting because even in unselected stocks, for example, here in the US, there's a shift slightly, and even in those, to somewhat resistance to varroa. So it's smaller than a selected stock, but there's still, so that shift is affecting the behavior of the mites also. That would be really interesting to investigate. So we noticed something. So we were looking at, just low varroa growth, and then we were looking at grooming behavior, and the colonies are grooming the most mites. So on the bottom boards, we noticed that some of the mites their legs were missing, or the shell of the shield was chewed out. So we were like looking at each other and thinking, what? So we decided, let's amplify this even more and let's select on mite biting. So we decided to start selecting on this trait. So typically you see the legs or the pedipalps are removed. But what really excites me is when you see the idiosoma chewed out like that. Maybe that's maybe a response to selection. So here's another beautiful picture that was photographed by Mark Gingrich. And this shows exactly what I was talking about. But here you look at this one, you look on the ventral side of the shield, you see this portion removed. So that's what I really like to see. And then you see the legs are removed. So the mites, when they get chewed, the leg gets chewed off, it bleeds out and then it desiccates and dies. So to select for traits, you need to have enough colonies. As I mentioned before, it takes a lot of time and resources. That's kind of the reason why there's really not a lot of bee breeders, is because it really does take a lot of work and time. And a lot of people don't have the time to do it. And it requires, an individual that is very observant and behavior and somebody who has intrinsic knowledge. There's a lot of factors that are involved for success. But one of them is just you need to have at least 100 colonies. So we have like 132, 34 colonies right now that are surviving. And so you want to have a nice population of bees. So you need you need to try and control the matings. So how do you do this? So we in our mating yards. So a lot of our queens are mating on the wing. They're not all instrumentally inseminated. And some of those queens that open mate and come back, they just, for whatever reason, hit the right constellation of drones and mated with them. And they had the right stuff. And so they become a breeder queen. So it's not always an instrumentally inseminated queen. So we think, through years of breeding in our area, we've kind of saturated this area with our type of genes. But yes, there's a beekeeper here, a beekeeper there, a so-called feral colony, that their drone population will be a part of our DCA. But most of the drones are from our hives. So we put drone combs in our top performers so we can produce more and hedge our bets that they mate with those only. And all the queens are marked, of course. So once the queen comes back and we like to let them lay for a month or more in a hive, and then we'll go in and we'll assess them for quality, how they're laying, the brood pattern, all that, and then we'll mark the queens. So how do you choose the parents? So you need to evaluate and keep good records. So your records is important. How do you control the matings? So you need to do this by using instrumental insemination or have a drone source flooding in your mating yard, as I talked about. So instrumental insemination is, just think of it as a tool. It's ideal to have that in a successful breeding program so you can control the mating of the queen. And you know exactly what drones, for example, they came from. So you need a trait that you can evaluate, something that's beneficial and something that is heritable. So you start with bees that have good traits in your area. So sometimes, like in some talks, I'll tell beekeepers that the best bees are in your area. You don't need to go to other parts of the country. You just need to work with the bees in your own backyard. So all these traits that I've talked about can be selected from the so-called whatever the worst stock is out there, whatever one wants to say is this bad stock. You can select all these traits out of it. So all these traits are already there. It's not like we're pulling something out of the magician's hat. It's already there. They're hardware. It's our job as breeders to select them out. So start with bees that are really doing well in your area. So I mentioned paint marking drones. So why do we paint mark the drones? So here is typically I'll paint mark them in the morning. And so we want to know the age of the drones and we want to know the source. So we call these patry lines, for example. So I have like this is the yellows. So this may be from say Queen A. And I might have blue, some B. So I know that the ages of the drones. So drones in literature, that they'll say the drones they're sexually mature at 12 days, for example. Well, most of the time, if you're bringing the drones into the lab to collect semen, very rarely do they have the right stuff at 12 days. So from the really age that well, they need to have ideal weather. So not, bright blue skies, warm temperatures, not a lot of wind. and able to fly, every day. So if they can fly every day up to 12 days and they age quicker, then the sperm migrates quicker, all that, et cetera. So yes, you may have luck then, but most often I wait until the drones are 20 days old. So when they're 20 days, that's when I bring them into the lab, and then we collect what we call germplasm or honeybee sperm. And we collect that into these glass straws, and then we inseminate the queens with them. So controlling knowing the source is important. So you can't just take unmarked drones and use them because you don't know what colony they came from. You need to know the source to make all the hard work well worth it. So this is a drone flight box that if you don't want to paint market drones, this is an option. So you have it's a triple deep blank straw, and the top box is basically a training center. So we have a queen excluder, we have the configuration, I'm sorry I don't have a picture of it, is you have a bottom board, and then you have a queen excluder so the drones can't escape. And then you have another deep, and then another queen excluder, and then you have the bees, the drones in the top here. This is what we call, this will be a flight block. So around, I don't know, 10 days, 8 days after they emerge, you just need to have that one queen excluder in the bottom board. So the second queen excluder is you put on when you're training them. So you take this inner cover off the top, and you put a queen excluder on. You kind of train them, so they fly around there. You take the lid off and put the queen excluder on, so they'll fly around. From around 2 o'clock, I'll do this and let them fly around until maybe 6, coming in the box itself. Then the day when around day 20, we have this improvised lid, or this hinge door with 8 mesh wire that we put on. So the drones, once you open it up on the day you want to collect the drones to bring into the lab to collect sperm, they're attracted to the light. So the sexually mature drones that are ready to go out and mate, they have like a photoperiod, or they're stimulated by the light also. So the more sexually mature they are, the more response they have to the light. So long story short, this is what it looks like. So you can stand in the shade of the tree. It might be, really hot, 85-degree day. So I like to get about 100 drones in there. So we did this a few times, but most often we paint them. So after a while, you'll get some workers that come in there because they start recruiting guards after you do this a few times. But it's really cool and it works well. So controlled mating, we use instrumental insemination, and basically we just cross the best with the best. So the highest mite-biter times the highest mite-biter. So we only select from those top performers. And this is just kind of a basic delineation of what's going on. So we collect the sperm cells. So here's one drone this is a classic example of a fully inverted drone. So his endophallus, the semen is actually at the end of the endophallus on this mucus bulb. So there's about 6 to 10 million sperm cells. So that's enough to fill the spermatheca, for example. The spermatheca has about 6 million sperm cells on average. So one can think, only one drone's worth of semen is really all the queen needs, even though she may made up with 70 individuals in rare cases, but most often, 15 to 20 individuals that becomes homogenized in her median and lateral oviducts. So I kind of think of the queen as kind of like a cement mixing truck before it migrates into the spermatheca. So within a couple of days, and then most of the sperm gets evicted out, and the workers eat it in the colony. So this is a glass syringe. We put this in the queen. So we'll either do, some cases we'll do single drone inseminations, or we'll do multiple drone inseminations depending on whatever we're wanting to do. So this is very important, this instrumental insemination technology for improving trait performance in honeybee stocks. And so it's very important. So here, ultimately in the end, we want to have a nice queen that's laying. So typically, about 5 days, we'll have eggs laid, and the success of the instrumental inseminated queens as opposed to the open May queens is no different. It all comes down to your technique and your preparations of the colonies and your queenering, as I mentioned earlier. If you've produced inferior queens, you're probably going to have more trouble inseminating the queens, or you'll have more problems with them down the line. So here is a beautiful mite-biter queen. She lived about 3 years. This was a 3-year-old queen, actually. And this is what you want, a really nice breeder queen. So we keep these colonies small. So this queen's kept in a single Langstroth hive. So she's not burning her candle on both ends. Yeah, you can put her in a production colony, and she's probably only going to last a year or 2 years. So we want to preserve, the genes, the sperm that she's producing in the eggs. So we try to keep the queens going as long as we can by keeping them in smaller colonies. You can also keep them in 5/5 nucleus colonies. We typically use deeps. We do that, deep nucleus colonies. So we're fortunate here in Indiana that our commercial beekeepers, here we have on the left here, this is Derek Shinnefield and his father Dave. So they're the 3rd generation commercial beekeepers, and they take mite-biters to California every year because they want to see how well they do in a commercial environment. So we've been fortunate. We've been working with them for years. When I joined Purdue University, I met Dave the first year because he worked with Greg Hunt in the beginning too when he started here. So we've been very fortunate to have a commercial beekeepers that support breeding. And we also have a breeder in Pennsylvania that produces mite-biter bees, and he goes to Florida every year, and he takes them back to Pennsylvania and disseminates them to whomever wants that type of stock in their bees. So it's really interesting. One time, he took an instrumentally inseminated queen to California 3 times. And I'm like, Dave, that's not what they're for. But he just wanted to see, because for him, longevity is important too, how long the queen's can live and things. So he took a high queen there. So interesting story. If you ever see him or want to talk to him, to have him ask him about that, it's interesting. So you can think of this breeding program, it's a hierarchical selection. So we're looking at grooming behavior, and then we measure the proportion of 2 mites. We eliminate those that show disease or did not control mite population growth. So it's not this simply, it takes a lot of finesse to, as I mentioned, to successfully breed bees. They're not, it sounds like we're doing single trait selection, but in reality, we're also manipulating other characters, like that chalk boot I mentioned. We eliminate that. So you can change other characters as you're selecting for certain traits, because we have what we call correlative trait responses. So certain traits bounce off other traits differently. So you need to pay attention to that and modulate and change your strategies on how your bees are responding. And the environment's a big factor too. It's very important to think about your environment while you're doing this selection. And keeping good records is essential for this. So bee breeding for me is like walking on a tightrope. It's not really a goal, it's a process. And you have to keep selecting. It's like a road with no ending. You have to keep selecting. You can't sit back and enjoy the fruits of your hard work and your selection without losing them. So that's what makes bee breeding difficult also, because honeybees mate on the wing, the high rate of polyandry, and they have what we call this recombination. So there's a high rate of recombination, the highest among any animal. on the planet. So there's a lot of factors that make it challenging. So for me, it's like walking this tightrope, and you have to constantly hang on and work as hard as you can, and hopefully things come out as you want. So I mentioned having too much of one trait. So here is a comb from one of our colonies. We don't select on VSH behavior, but every so often we have one that does this. So it's, there's a bee going by and she detects some anomaly, and another bee decides, oh, it's okay, and another bee starts. So all the ones that have the raised rims here, those are going to be recapped. So you have one bee, so there's one gene for uncapping, there's another gene for recapping, etc. So this is VSH gone wild, for example. So we can lose traits pretty quickly without selection. So a lot of problems that occurs, beekeepers are buying inseminated queens from places, and then they open mate on the wing. And then so you can see now some of these traits, again, can be lost quicker than others. So this is just kind of to show you that through time, through 10 generations, you can lose some of these traits. So the nice thing is, mite biting is a heritable trait. And this is a trait that hopefully more people now are having, because we're one big factor to think about is there's resistance to Amitraz occurring. I'm hearing about it all the time. So breeding is something that will become more popular. So as I'm talking now, there is more interest in honeybee breeding. So it's a heritable trait. So colonies that chew more mites also have less mites. So you have fewer mites in a colony. That means less problems. You have less propensity for virus outbreaks and things. So keeping the mite, so there's different ways of reducing the mite population. So you could do it with VSH type bees, you can do it with grooming, you can do it with mite biting. So it's just a matter of selecting on those traits. So some challenges with this mite biting assays, you need to have mites. So since we've been selecting on this, we do have fewer mites. And in the springtime, especially like when we put our sticky boards in and we want to check, we don't see a lot of mites. And why is that? Well, one factor is most of the mites, I mean, 90% or more, especially in the early spring, they're going to be in a brood. So a lot of the mites are there. There's also poor repeatability sometimes. Like we'll have certain moments when if there's a high honey flow, for example, there seems to be a less grooming going on during that time. And then, as I mentioned, correlated traits, different effects of different traits. But the history of the colony is a big factor too. So did your colony swarm? Did you kill the queen accidentally? Did your colony supersede the queen? So any loss of queen event is going to put a brood, it's going to cause a brood break. So a brood break will reduce the number of varroa that can successfully reproduce. Now, some mites are just for whatever reason not successful at reproducing. She invades a cell and she doesn't produce a family for whatever reason. So the history is very important. A few years ago, there was a paper published about bees biting and 2-heptanone and a correlation between being paralyzed. So we used to think as 2-heptanone as an alarm pheromone. So it has alarm pheromone components, but it also turns out that it causes paralysis of the mites, or say of a wax moth larva. So the bees bite for a reason. So that was exciting. This is open access. If you Google this, you can find and read about that. So that was exciting to see that. I think it's been about 10 years ago now when this was published. So this groove here, you see, this is where the 2-heptanone comes down. So when a bee bites you, she releases that 2-heptanone. So it also tracks bees' defensive behavior, stinging behavior. So you see this. So both the paired mandibles have these grooves. So I'm going to talk more about a recent study. We've done 4 different comparison studies with stock that's typically purchased by commercial beekeepers, sideliners, and hobbyists. We looked at Carniolans and Caucasian stocks, and the same thing across the board. Overall, the Purdue, the Indiana mite-biters outperformed those non-selected stocks. So there was less winter mortality, etc. So the colonies performed well. There's fewer mites in the selected stock versus the non-selected stock. So this is the results of an Indiana mite-biter versus a tie-in stock. So there's different regions in the chromosomes that represent different traits. So VSH, I think there's like 6 or 7 genes associated. And with mite biting, we don't really know anything. We don't even know, is it recessive? Is it dominant? All we know is we had a theory of one gene. Of course, there's probably other genes, so this needs to be investigated. So Nerexin-1 was a gene that we had our eyes on. So we have this paper published on this. It's also open access if anybody wants to read about this in great detail. It's out there for you. So basically, let me move this. So 18 colonies were split in June, mid-June, in one apiary, and then we sampled in July 10, only 25 days since the split on October 11th. And then we assessed for mite drop, proportion of chewed mites, and we also looked at the amount of virus that was floating around in the bee's blood, in the virus titers. So we looked at grooming behavior and mite levels. So we found that, trying to get this thing out of the way, there we go. So we looked at sample dates of the queen source, mite drop, and chewed. So the Italians, if you look 10 July, so they had more mites, and the Purdue's had fewer. And then you look in October, the Italian mite drop was more significant. But what's even greater is this: you look at the percent chewed. So there's a response to selection. So colonies that aren't selected for this trait, for example, express much less of that, but they do express it. So again, all unselected stock out there, and they're expressing it at a higher level than, say, they did, in the early 80s, before, because there's a presence of varroa. So there's a shift that's occurring. So we're only trying to speed that up through selection with breeding. So the breeding, all the honeybee breeding programs out there are just simply trying to speed up what will probably ultimately happen in many, many years. And the major viruses we detected was DWV, black and queen cell virus, and lake cyanide virus. So DWV correlated with the mite levels. There's a big correlation. Winter survival as of February, the Purdue's were at 77% and the Italians at 22%. And this, again, is similar to what we found in our other comparison studies. So the results, you look at the varroa population growth between the Italian and the Indiana mite-biter. Again, it's like night and day here. And then you look at the proportion of mutilated mites. So again, the selected stock is showing a much higher proportion against the unselected stock. But what was most compelling in this was Nerexin gene expression correlated with the proportion of chewed mites. So the Nerexin shows there's a higher synaptic response in the brain of the honeybee. So this is in the brain. And so the bees that were grooming more had a higher expression of this, but also they were the ones that were chewing the most mites. So that was really exciting. So looking ahead, we're going to be looking at stock comparisons, teaching queen rearing and instrumental insemination. So we teach these classes, 2 advanced courses every year here at Purdue. And then we're expanding infrastructure and then queen production and selection and collaborative efforts with micro breeders. So there's many breeders here in Indiana, for example, that are interested in other states, and then using mite-biter stock in commercial operation. And we're also incorporating genomic selection into our phenotypic breeding program. So for years we did phenotypic selection, and now we're going to use genomic selection. So with genomic selection, you need to have many individuals, and ideally you want to identify as many markers also associated with different genes or traits that you're interested in. So we have, for example, recently we got we identified a mutant eye color in drones. So we have different variations of, say, white eyes, there's purple eyes, or chartreuse, there's kind of a yellow or red. So we identified one of these genes, and we will have this published probably by this summer or early fall. So that'll be exciting to see. So bees are fighting back. It's just one step at a time. So as bee breeders, we can amplify these traits. In Indiana, bee breeding is taken very seriously. So we have the Indian Queen Breeders Association was inspired by our breeding program, and they started working together, and they come to our lab every year, and they get mite-biter stock, and they bring their stock, and we cross them with survivor stocks, and then they disseminate to other breeders or other states. So we kind of funnel our queens through the IQBA. And then wrapping up here, just a few people to thank here, but above all, Greg Hunt, my great friend and mentors. So he was fantastic to work with through the years. And with that, I think I'm all finished. Thank you very much.
Well, thank you, Krispn. I mean, that was fantastic. Really,
Thank
really
you.
good. Yeah, thank you so much.
Yeah, there's so much talk about VSH now. It's like there's other traits also out there. So we just need to get more people interested in this too. I mean,
yeah, you're absolutely right. All of the thank yous are coming in on the chats. I hope you can see them, but
I'm sorry.
yeah, it was fantastic.
Maybe if I open this.
Yeah, no, that's okay.
It's I kind of feel like a lone wolf.
Yeah.
So for a long time, we had hundreds of people coming over that got our, from 11 to about 2018, hundreds of breeder queens went out the door and went here, went there, and the last few years is kind of people are saying this is trait can't be selected for. I mean, it's absurd. I mean, we've proven that this is a heritable trait that can be selected for, just like VSH. It's just some individuals that aren't selecting on this trait want to highlight their stuff. So that's all. It's just important trait also that we can work with.
Well, it was, I mean, bless your heart for doing it. Thank you. It was fantastic. We have a few questions. Krispn, do you have time to take a few questions?
Yeah. Yes.
All right. So I'll give these, I'll go through these in the order that they came in.
Okay.
One was, how much time do you estimate is spent in a season to do all the grooming tests in each colony? They said they're trying to understand the feasibility to do this in say, a regular operation.
Yeah. So, yeah, that's a good question. I say the bringing in, just collecting the samples in a day's time. I mean, you're gonna spend 8 hours collecting, removing the mites from the glass slides, placing them on and investigating the legs. So you could spend easily 8 hours a day. So, say 8 hours will our underground our tech will spend on it. But in the beginning, I did do this myself. I also collected the mice and looked at them under a microscope. So you could spend, 40 hours, depending on the number of colonies you have, 40 hours a week just looking at this trait. But again, you only want to, ideally, if you can do it have 3 time points, that would be great. But most often we only do it in the spring and the fall. So so just looking at the mites, the physical mites and moving them, that's not including the instrumental insemination, the rearing of the queens. So it's a full-time job for if somebody seriously wants to do it. Like VSH bees, for example, people are doing this. There's not a lot, but it's a full-time job. So you're not just looking at the characters, but you also have to do the queen rearing and the instrumental insemination. So I can't tell you exactly how many hours, but yeah, you could spend, certain times a year, 40 hours a week, no problem. But certain months, yeah, might be a slower month, like July, for example, here is a slower month as far as.
Yeah. And I would imagine that you also, you need to have a microscope and slides and be able to,
Yes.
so you need the equipment to be able to look at them.
Yeah, that's a 15x microscope. You can buy a cheap one off of eBay. Amscope makes a really wonderful microscope and they're pretty cheap because beekeepers, most of us are pretty thrifty. And so, yeah, most of them are pretty cheap you can buy. And it's seems overwhelming at first, but once you start selecting on this trait, you figure it's just like riding a bike or any difficult thing. After a while, it becomes easy. Playing the piano, after playing the piano, it becomes there's muscle memory, so to speak. So don't be discouraged. I mean, I'm encouraging if anything, if I can just inspire one person out there to start selecting on this trait also, that would be awesome. But again,
Yeah.
you need to have at least 100 colonies and you don't have to be a geneticist or no, you just got to have intrinsic knowledge. Some of the best beekeepers I've ever met are beekeepers that don't read a lot of books and they just are really good. They're born with that innate ability. So
Yeah.
that's what we need is more bee breeders. We got enough queen rearers. So
Yeah.
yes.
That's true. So one other question, which is a follow-up to that, is in your drone saturated areas, what percentage of open mated queens carry the grooming trait versus your inseminated queens?
Yeah, that's a great question. So that now in some cases, let's say out of 10 queens, maybe like 6 would have pretty good stuff. And then out of 6, maybe 1 or 2 would have the right stuff. But out of 100, yeah. So if you just look at out of 10 to keep it simple. So not all the queens, even with instrumental insemination, for whatever reason, when you do certain crosses, so we'll do what we call reciprocal crosses. We'll cross the drone with the queen with the drone. And in some cases, those crosses don't work. They don't have the right stuff. And then when you bring in another source, like years ago when we brought in VSH bees, they were a little defensive all the way, up to the F3. So if you F1, let's say out of 10 queens, maybe 4 of those were a little, they're a little defensive, a little grumpy. But the key is to push it to the next generation and then use the select gentlest so you can weed out that defensive behavior. Because there's some word going around, well, if you have mite biting bees, you got mean bees. That's not true at all. If we got bees here for years, we teach beekeepers. We have beekeepers visiting, their people are getting stung up, and the colonies also produce a lot of honey in this area. So it's a great trait. And so yes, some of those open mated queens do not have the right stuff. So we don't use those queens.
Yeah, so if I'm hearing you right, and if I understood your presentation, the aggressiveness thing is uh, not a characteristic of a mite biting queen. It's the grooming behavior that is that
Yeah,
right?
there seems to be Ernesto Guzman also is looking at grooming behavior, and that question's been on. It's like it seems like they're not like highly defensive, but they're just a little more temperamental.
Mm.
So it just it seemed, but we don't. He doesn't really know exactly. In our experience with ours, as I mentioned, sometimes if you have, especially if you bring in an outcross or a top cross, you want to bring something different in and just test, and that's done in a controlled yard. And sometimes like those VSH early bees, we had some, but we were able to weed that out. You just have to push the trait further. You can't say, at the first generation that, oh, they're mean bees, they're no good. No, no, no. You got to push. If you produce 10 queens, you're going to get one or two that are not defensive. You might get intermediates, and you might get one that's a little testy. And for whatever reason, it may be even with a simple cross with a Carnica or Caucasian, you'll get defensive alleles for whatever they express themselves. So as
Right.
a breeder, you have to just tease those out. So if you have something really good going, you can select that defensive behavior out of your bees.
Yeah, good. So the next question is, does the mite biting kill the mites, or is there any evidence that the bees can contact the viruses orally by biting the mites? That's a good question. Yeah,
Yeah, that's a great question. So many great questions now. Yeah, so in theory, I maybe somebody knows more about this than me, but I think if the mite gets bitten and they bleed out kind of like a spider, I mean, it's like a hydraulic system. I mean, it seems like, yeah, if the legs are removed, the mite dies, but a lot of times when they remove the act of uh, grooming and mite biting, the mite is also dropped on the bottom board. It's not present in the colony. But it'd be interesting to, pull a leg off a mite and see if it can survive.
yeah,
Nobody, I don't know if it's done that, but yeah, and the virus get them through the mite. Yes. So I don't, I don't really know the answer to that, but it seems like it's quite possible. If a freshly killed mite has a lot of virus in it, and
yeah,
because the bees more likely eat some of the blood of the mites also. So
right.
it's possible, yeah, to transmit the virus that way. I wouldn't. That's an excellent question. I really don't know the answer to it, but in theory, yes.
So a future maybe study on that,
Yes.
but
Yeah, there's so many unknowns.
yeah. So another question is, since instrumentally inseminated queens never take their mating flights, do you think this has an effect on the queen, such as increased swarming or other issues you've seen with them not having ever gone on their natural mating flight?
No, no, there's no difference. I mean, if you have really great technique and you have superior queen rearing skills and advanced beekeeping skills, I mean, it takes a lot of finesse to do all this.
Yeah,
And so if you instrumentally inseminate a queen successfully, she's going to perform just as well as an open mate queen. So if there's a more propensity for swarming, that would be a genetic thing. So yes, some bees swarm more than other bees are more swarmy, like Carnica bees at their purest state. I mean, a lot of the stuff that's Carniola and Caucasian that's out there being sold and available to beekeepers isn't truly pure and Italian anything. They're mongrels, they're mutts. So, but that's a good thing, and so that's what's made our bees a little more uh, healthier and
yeah,
with all these multiple. But yeah, so there's really no, it doesn't seem to be a more swarming or anything associated with the queen. But something I might add, instrumentally inseminated queen will attempt to take a mating flight. So if sometimes when I'm doing single drone inseminations, if I don't put queen excluder on that nucleus colony and don't clip the queen's wings, the queen will fly out because she doesn't feel like she has enough stuff. So she can sense, how much sperm is in her. She has receptors in her abdomen that say, hey, I got enough sperm, or hey, I don't have enough sperm. So, for example, a queen that goes on a mating flight and it only mates with 2 drones, she may go out on another mating flight the next day. So she'll have 2 mating signs, for example. She'll come back from day 1 mating, she'll have a mating sign. It's just a plug, it's the endophallus. It breaks off from the drone and is left in to the queen. It's a semen plug, if you think of it that way. So the queen that will be removed overnight by the bees and eaten, and some of it removed from the colony. But then she'll go out on a second mating flight because she feels like she needs more sperm. So II queens can and do have a will to go on a mating flight. So you have to make sure you have queen excluder on all the mating nuclei, and we clip one wing to the very edge of the wing a little bit to keep her from flying, but she still can crawl out. So you want to put the queen excluder on, and the queens are inseminated 2 times, or I'm sorry, they're gassed 2 times with carbon dioxide. So we narcotize them twice. So this initiates egg laying, oviposition pretty quick. So within, 3 to 5 days, you're going to have your first eggs laid. If it's, warm, nice weather, etc.
Yeah, and one downside of doing this through open mating is there's a risk the queen doesn't come back. And so,
Yes, yeah, we have one of our mating yards, we have a big pond nearby, and what's around big ponds? Dragonflies. They,
yeah,
yes, every once in a while, certain queens don't make it back. But last year was really good. I mean, we had about 90% take. I mean, that's fantastic. I'm happy if I can just get
wow,
60, 70%, but typically we get around 80% take. But
yeah,
last year was exceptional.
great. Well, another question, is there any way, and this is, I think, a good one to address, is there any way a backyard beekeeper with just a few hives can support a breeding program?
Yeah, it's an excellent question. Years ago, there was another organization that was inspired by our breeding program that is still out there, but we don't really work with them that much. I got them on this slide here. It's called the Heartland Honeybee Breeders Cooperative. So a few years ago, these beekeepers from 7 states, some of them had just a few colonies, so that they formed this co-op where you have many, not just the bee breeders, but even hobbyist beekeepers working together. So we were disseminating germplasm, and we were also disseminating information on how to successfully have a breeding program, or how to inseminate queens, all this stuff. So we call it like the insemination fest. So they would come to our lab every year, 7 states, and they would get mite-biter stock, and then they would be disseminated throughout all the states. So we did that all the way up until 2020 with them, and now they kind of are off doing something else. So right now, most of the stuff is funneled through the Indiana queen breeders, and then every so often a university or a breeder may request some queens from us.
Yeah, so another question is, do you assess SMR VSH in your high grooming colonies? I'm curious if you find any overlap.
Yeah, no, and with the beginning, we got some of this VSA, but we never really, we don't select on that trait.
Huh.
But every so often we get a colony, even now, I mean, every so often now, of 100 hives, we might have 1 or 2. Once in a while, they show this uncapping and recapping. I'm sure some of you have seen this in your colonies, but you've never purchased the so-called VSHB. So again, these traits are out there, and sometimes, you get all these trait values that come together even in the open mated world to where they produce a stock that expresses that trait more.
Yeah. So another, which is a good question, and I think it's probably on people's minds, has your program been successful enough to replace traditional mite treatments?
No, now I would say I'm not against treating for mites. We don't treat ours unless the mites overwhelm the colony. So even in every breeding colony, every breeding population, I mean, there's going to be, for whatever reason, there's colonies that can have many mites and they just know how to deal with them, but there's also colonies that still have problems. So those colonies will treat. So we typically use oxalic acid in our colonies to treat those.
Great. Okay, so a couple of questions just on my mind. I'm sure they haven't, others are thinking this, they just haven't been typed in yet, is, so let's say that, let's say that,
I
what's that?
forgot to mention thiamol is another product that we
yeah.
use that works well.
Yeah, good. So if somebody wants to purchase a mated queen from you guys, the inseminated queen, what's the process for that?
Yeah, so here at Purdue, we don't have a lot of time to produce lots of breeder queens or lots of open mated queens. We just don't have the resources or the time for that. So we funnel a lot of our stuff out to other breeders, for example, the Indiana queen breeders. And so we'll tell folks to go there, but we do offer a few queens every year to certain breeders. So the key is just email me and I can tell you what type of availability there is. But we're not able to produce, we're not in the queen or, the queen sale business. It's just, we don't have enough time for it. So that's why if we can get more breeders breeding this trait, instead of just going down a single path, which is VSH, I mean, let's look at other paths. I mean, there's grooming behavior. We know that's a heritable trait. There's mite biting. Yeah, it's difficult too. Let's have more people investigate this. And then why not, have bees you can work with, you could have VSH bees and you could cross them with mite biters. These are things that could be investigated, or you can have two different. It's just important not to have all your eggs in one basket. We just have to the take home message also is there's other traits out there, and this is a trait that we've done quite a bit of work on and successfully demonstrated that it's a heritable trait.
Right, right. Well, it was great. Krispn, thank you
Thank you
so
very
much,
much,
Dr.
Scott.
Given. Yeah, it was
Yeah.
fantastic.